Hello, welcome to Vedial Vagaparai. In this video, we are going to see a GATE 2025 question, which is based on partial hydrolysis of peptides. This particular method is very popular method to identify the peptide sequence. Whenever we are talking about peptide sequence, we know we use Edmonds method or the Sanger's method to sequence the amino acids of a peptide sequence from the N-terminal end, and we use enzymatic method to sequence the protein chain from the C-terminal end. If you are interested in understanding about Edmonds method, Sanger's method, please see the videos that we have already posted so that they will be very helpful for you. In this particular video, we are going to see how partial hydrolysis is very helpful in identifying the peptide sequence. This is one of the earliest methods of identifying peptide sequence, well before the identification of Edmonds method or other modern methods. And Sanger and his team used this method to identify and sequence the amino acids in insulin. So let's see the question first. Partial hydrolysis of a pentapeptide yields all possible tripeptides and dipeptides. The dipeptides that are obtained upon hydrolysis are given below. The total number of tripeptides obtained that contain alanine as one of the amino acids. So in this particular question, we see that it is a pentapeptide, and they are saying there are different possible tripeptide and dipeptide sequences were formed, or sorry, fragments were formed, and they have given the fragments of dipeptides that were obtained in this particular question. So using the fragments of dipeptides, we can propose the possible sequence of pentapeptides, and based on the pentapeptide sequence, we can propose the possible tripeptide fragments. So in this particular question, first what we will do is, using this data, we will find the pentapeptide sequence, and from the pentapeptide sequence, we will find the fragments of how many tripeptide fragments can be obtained that contain alanine as the residue. So let's see the question. So in this particular question, as I told you, we have 4 fragments of dipeptides that are given. So in this 4 fragments of dipeptide that are given, we have valine, alanine, glutamine, histidine, phenylalanine, valine, alanine, glutamine. So I have used same color for the amino acids when they are repeated, so that we know how we can go about doing a permutation and combinations. So let me begin with valine and alanine, which is the proposed sequence at this end. So after valine, we have alanine attached to it. Then we also see another sequence where alanine is attached to glutamine. So definitely next to this sequence, this is the sequence that is expected. So we will place that sequence next. Next, what we will see is glutamine is bonded to histidine. And always we must remember why we are doing this is, um, amino acids are arranged in a peptide from the N-terminal end to the C-terminal end. So that is what I am doing. So assuming that valine is the N-terminal end, I am just building the fragments up to the C-terminal end. So first I started with valine, then I have the alanine segment, then the third segment is a glutamine histidine segment. So now what I see is phenylalanine is actually before valine. So definitely valine is not the N-terminal amino acid. Instead, it is phenylalanine. So now when we have arranged these fragments, we see the amino acids are common in between. So when we merge these common fragments, we will get the desired pentapeptide. So let us see this. So phenylalanine, valine, then valine alanine, then alanine glutamine, then glutamine histidine. So what we see here is the pentapeptide, which is phenylalanine, valine, alanine, glutamine, and histidine. So as a first part, we have found out the pentapeptide sequence using the dipeptide fragments that are given here. This is how scientists used the partial hydrolysis to arrive at the peptide sequence. Now that we have found the peptide sequence, the question is, what is the number of tripeptides that can be got, which will contain at least one alanine residue? So we know pretty well we have to fragment around alanine so that I get a tripeptide. I can break it here, I can break it here, I can break out the end amino acids to get a tripeptide in between. So that is what we're going to do. So the first is the breaking of the phenyl-valine and alanine. Then we have valine, alanine, and glutamine. And the third is alanine, glutamine, and histidine. So these are the only three ways we can have alanine as a residue in a tripeptide. So this is how we fragment a pentapeptide, and we arrive at the number of possible tripeptides that can be obtained from this particular fragment. So the answer for this particular question is 3. I hope you understood. Kindly subscribe for more videos. Thank you.